My pleasure to introduce Dalton Hagi, who is finishing up his master's degree in Dr. Patricia Klein's lab. He majored in crop science for his bachelor's degree at the University of Illinois, Urbana-Champaign, from where he graduated in 2019, came straight down to Texas A&M, joining the Mets department. He wasn't born in Texas, but he got here as soon as he could. Dr. Klein's lab has been working on pecans, not pecans for you northerners, and is presenting one of his projects looking at the morphology of pecan nuts.
All right, thanks for that introduction. So welcome everyone, thanks for coming to today. All right, so I hope you guys will enjoy this presentation of one of the projects I've been working on my time here, looking at the QTL analysis of pecan fruit morphological traits. Let's get started with some background here. Pecan is included in the genus Caria, otherwise known as the hickories, which contain 18 species, some being native to Asia and others being native to Northern America. Pecan is one of the species that is native to North America, shown here in its native range, shaded here in red on the top. But commercially, it's produced in a much wider region, shaded here in yellow on the bottom, ranging from California to Georgia. And it's also produced in other countries throughout the world. We usually think of it here mostly at Texas A&M for its nutritional aspects and the edible kernel. But in general, the hickories as a whole are known to be used for ornamental purposes as well, being used commonly in furniture building, smoking quality for Texas barbecue, and even landscaping. But coming back to the nutritional aspects and pecan specifically, they're being increasingly found to be great sources of antioxidants and healthy fats, which have been shown in other studies to help mitigate inflammation and reduce the risk of cardiovascular disease. So my project talks about nut morphology, but let's go back to the beginning of that process, which starts with flowering. And so pecans are monoecious, and so they have separate male and female flowers on the same tree. But to make things a little bit more complicated, they also display what's known as dichotomy, which is a term used to describe the difference in maturity times of the separate male and female flowers on a tree. And so you have some trees that have male flowers mature and shed pollen before the female flowers on that same tree are receptive. And those would cross with the opposite of that, which is trees that have female flowers produce and are receptive first before the male flowers on that tree shed pollen. And this is an evolutionary mechanism designed to maximize outcrossing and minimize the selfing of a tree as they display high levels of inbreeding depression. And usually, and pecan and other tree nut species in general are usually outcrossing and wind pollinated. This aspect of dichotomy makes the breeding of pecan a lot more challenging than, say, other annual crops. But once these female flowers are fertilized at the terminal ends of the branches, we start to form these tiny nutlet clusters that can range anywhere from 2 to 6 per cluster. But we really are looking for around 3 to 4 per cluster, because that's the sweet spot between having enough nuts on the tree to make a good yield in that year, but not decreasing on the quality of those nuts. Because if we had 6 nuts per cluster, say all throughout the tree, that would really just overload that tree and stress it out. And there probably would not be enough water in the growing season to really efficiently fill those nuts out and make a good crop. So to look at this and whole timeline at like a 3,000-foot view, we're looking at bud break around this time of the year where the trees are coming out of dormancy in mid to late March throughout April. And then right at the end of that, we have those protandrous and protogynous trees crossing with each other. Fertilization happens, and we start to move into the 2 phases of nut development beginning in May. And the first phase goes from around May to August, the middle of August, where we're looking at the trees just focusing on steadily increasing the size of those nuts and really creating the outer shell of that nut. And then once the end of summer comes around and we move to the middle of August, the trees start shifting their focus on actually filling those shells that they created. And so the actual size of the nut is finalized at that point, and it's looking at filling that. The second phase can really differ in time depending on genetic background. So as I mentioned, the native ranges from Illinois to Texas, and so there's quite a difference in latitude there. And the northern material, as you would guess, matures much quicker than the southern material. And so a great example of this would be the cultivar Pawnee, which was released by the USDA pecan breeding program like several years back. And it's known to mature around and be harvestable around September, the end of September. Whereas more native material to Texas is usually harvested around the beginning of November. And so you're looking at a difference of about a month's worth of time in terms of harvesting between genetic backgrounds. This can affect the prices received at market, as the earlier harvested material usually gets higher prices as the supply is lower. But once you harvest these, you can keep them in shell for multiple years if you keep them in cool storage. So what else makes pecan so viable in terms of a tree nut species and using it as an agronomical crop? Well, for one, a lot of the hickories have a bitter or astringent flavor. And so you're looking at negating a lot of the use in those from the get-go. But there are a couple other species within the hickories that have a good flavor as pecan would, one of those being shagbark hickory. But these also contain challenges that pecan doesn't really face, and that has to do with the shell architecture. And so as you see here where these arrows are pointing, these extrusions of the shell that come in the grooves of the kernel, as well as this middle plate here that separates the two kernel halves, is a direct extension of that hard woody shell material. And so when you compare that with combine that with the deep cleft that you see in the actual morphology of the kernel, it really traps those kernels in the shell and makes it hard to quickly extract those kernels. Whereas we don't really see that in pecan. If you look here, there's an abscission layer really, and we're looking at a loose packing material in those same areas as compared to the shagbark hickory. And they also have an absence of a deep cleft in their kernel. And so once you open that nut, open that shell, it's really much more easy for the kernels to actually fall out of those shells, and gives them their viability as a high agronomical crop on a commercial level. So the relevance of this project, like other species of other common annual crops like tomato, we see a great deal of variability within the shape and size of pecans. They can range from tiny to rather large, as well as the shape of these pecans do have an effect on the end product and quality of these. So we see ranges from more oblong or blocky shapes like Wichita to very spherical in shape like Shoshone. And these do have aspects and effects on harvesting efficiency, which Gerald Sparks kind of mentions in his book that he published in 1992, which he did state some important reasons as to why nut shape is important in pecan. Some of them being consumers prefer a blocky shape, which would be more like you see here in Cape Fear, where it's kind of just a generic shape. A lot of earlier successful cultivars had this similar shape. Very elongated nuts, more like Mohawk here, tend to need ideal conditions to really fill out well. And then most importantly out of these points is that round nuts like Shoshone here have a greater chance to sit crosswise in the cracker when it comes to extracting, leading to breaking kernel halves. So not a lot of work at all really has been done in looking at the genetic control of morphology of pecans. And so we had the opportunity to really get started on that, working with the USDA pecan breeding station just outside Call Station here. We have a genetic mapping population that's just now starting to come into sexual maturity within the last few years. And so we wanted to set up a protocol to quickly screen those for morphology data and see if there's any traits that are segregating pertaining to morphology within this population, and seeing which regions of the genome are responsible. And so getting to the actual mapping population, term CS map for College Station, it contains 151 F1 progeny between reciprocal crosses of two common cultivars, Elliott and VC168, whose nuts are shown here for reference, but Elliott is a much smaller size nut and has a very spherical shape, whereas the VC168 has a much more oblong or blocky shape, and it's moderate to large in size. And so these progeny were created in 2007 and 2008, and in 2019 was really the first collection that they finally started producing the pecans themselves. And so it's just an example of the extremely long juvenility period in this crop, which presents its own challenges in itself. But we did want to just see what we could find with some of the first trees fruiting in this population. And so which were collected in 2019 and 2020. And so in 2019, only 45 trees fruited, which I know doesn't sound like a lot, but we wanted, like I said, just to get our feet wet and start with this process. Everything was collected by the USDA, and they were looking at collecting and measuring 5 nuts per tree. In 2020, I got to go out there and do everything and collect everything using the boom lift here, getting up in the canopy. We had an increase in trees, looking at 55 that year. And so I collected 15 for the USDA's annual measurements, as well as an additional 32 for my own measurements back here at lab. And so between both years, we're looking at 65 trees here, with 35 doing so in fruiting in both 2019 and 2020, which will become important later on for looking at some of these quality and morphological aspects. In 2021, as it's been the theme throughout everyone's projects here, that year was kind of troublesome with weather, and that crazy freeze that we had come through that year in February, I think damaged the floral buds that year so harshly that there was really no pollination that went on that year. And so there was really no nut set that year. And so it would have been the 3rd year of data. It wasn't able to be collected. But for the 2 years that we had in 2019 and 2020, the phenotyping was done, like I mentioned, by the USDA in 2019, looking at their typical nut quality measurements like length, width, height, volume, and how well those nuts were filled out with percentage of fill. And in 2020, I replicated alongside their measurements that year, doing all their typical quality measurements, as well as looking at new aspects that really hone in more on the overall shape, and doing so in a more favorable to a high throughput system, doing this digitally. And so I would have the nuts scanned through a scanner, which would generate this image for me. And then I could throw that into different analyzing softwares like Tomato Analyzer, which was developed, an imaging software used to look at the morphology of tomato, developed by the Vanderknapp lab from Ohio, who's done a bunch of great work in tomato morphology. And also using open source softwares like ImageJ. And together with those, looking at things like the distal angle of the nut, or how pointed the ends of the nuts were, the degree of how circular they were, looking at interior things like shell thickness, as well as the depth and the width of these grooves here that you, or the kernel grooves, as that's been one of the breeding strategies or targets of pecan at the USDA in the past, is these have direct effects on end product quality as well. And so after collecting all this data, running it through actual QTL software, use our QTL in RStudio, looking at the scan one function, which is a single QTL model. Anything that popped back up as significant was rerun using 1,000 permutations, just to ensure that these are legit QTL. And then anything I report here is surpassing the 95% cutoff value. And so getting into results. In 2019, just looking at this year by itself, no really final conclusions were made, as nothing really popped back up when just looking at this year by itself. And I think that has to do with just 45 trees still wasn't enough of a threshold to really hone in on the segregation of these traits. But regardless of 2019 and 2020, the data collected in those years shown a normal distribution, as I show some of the traits here, just for a picture. But then looking at everything, all phenotypes collected, and looking at what patterns were really thrown in through this data, doing a principal components analysis, it really gave the data a spread into 4 separate quadrants, separating the nuts based on overall shape and overall size, which is, if we look here, the larger size pecans could be found on the right side of this principal component, with the oblong or blocky shaped nuts being typically found in this top quadrant, the large spherical nuts being typically found in this bottom quadrant, and then we move over to the smaller size nuts, which show the same pattern, with oblong being on top and spherical being found in the bottom quadrant. So this is just to show that we had a pretty good spread of phenotypes saw in this, or observed in this data set, which led to more promising results for 2020, as we had that increase in trees, as well as doing some new measurements with the morphology imaging softwares. Found several QTL pertaining to morphology. Getting into those a little bit more, nut height and kernel weight were found at the same locus on chromosome 11 of Elliott. explaining 27% of the variance, but I do want to state that we know that these are inflated values since we're dealing with a small number of individuals, but the positions in the QTL are still existent. But we're looking at perhaps a pleiotropic QTL here where we're dealing with two separate traits being controlled by a similar QTL. Getting more into the shape aspect, as I mentioned, looking at the distal angle using that tomato analyzer software, how pointed the ends of these nuts are, showing a QTL segregating on chromosome 3 of Elliot, as well as moving toward the opposite end of the nut, looking here at the proximal end and how really asymmetrical those nuts are, how much mass is found on the top half of the nut versus the bottom, showing a QTL for chromosome 3 of the other parent on VC168. Shell thickness was proving to segregate in this population pretty strongly, as there's two separate QTL found from both parents, one on chromosome 1 of VC168, and another strong QTL here on chromosome 5 of Elliot, which there was a twofold difference throughout the population in terms of range of this phenotype. Looking at some very thin shelled nuts here that had an average of around 0.67, all the way up to double that value, which is one of the thickest shelled here, looking at an average of 1.4 millimeters. And while it doesn't sound like a lot on this scale, it does make quite a difference, as you could typically extract or peel apart the shell of this thin shell here with your hands, but you need a nut, like you need an actual hand cracker to shell these thick shelled nuts here. And then the last QTL specifically for 2020 data only was looking at the coronal groove width, which shown a pretty strong QTL on chromosome 10 of VC168 with a log of 5.13. And I did wonder if perhaps the validity of this phenotype, as maybe coronal groove width was just directly related to the overall width of the nut, as those might have wider groove widths based off of they have more size within that shell to spare. But it didn't seem to be the case, and I'll touch on that in a bit. But then pertaining to those 35 trees that I did mention, fruited in both 2019 and 2020, I did want to look into see if shape was relatively stable throughout the years so that we could combine trees going forward to increase our number of trees involved in analyses. And so I did a two means test with just the basic external trait measurements for those, like length, width, and height. And I did fail to reject that the means were different or same, meaning they were the same from year to year. And so I did go ahead and combine those and run those through a QTL analysis as well with the years combined, and did find some more QTL for length, width, and overall volume. So getting to length and width, these were stemming from relatively the same position on chromosome 5 of Elliot, and were in fact actually just downstream of that QTL found for shell thickness. And so we're seeing a general region here. These QTL positions did show up for length and width when analyzing 2020 by itself. It's just that I think with that lower number of progeny used, it wasn't enough statistical power to surpass a 95% threshold. But we did see some stability in the positions of those QTL. And then getting to overall size and volume, we're looking at the same location on chromosome 5 of Elliot, which makes sense here since we're looking at length and width being two of the three factors that calculate volume besides height. And we see that in the fact that the interval for these phenotypes are containing both regions for width and length, which is most likely why we see these two separate little peaks here. Looking at correlations in this data set, some that were worthy of mentioning, kernel weight had a slight positive correlation with the ovoid shape characteristic, which is just another term for that proximal blockiness or that upside down triangle shape. Shell thickness, circled these. Shell thickness had a slight positive correlation with height and width, but not length. Nut length was less correlated with height compared to width, which I thought was interesting and kind of just backed up the fact that we found QTL positions for length and width right next to each other, but height was on a completely separate chromosome. And then lastly, as I mentioned, being suspicious of that coronal groove width relating to nut width overall, but as we see here, there's actually not really any relationship pertaining to that. And so that did lead me to believe that this is a true phenotype that we're seeing. So as you might have noticed, a distal end of chromosome 5 seems to be a real hotspot for a lot of these morphometric traits like shell thickness, nut width, and nut length, which ties into volume. And when looking through the literature, this seemed to be a lot of the case for other morphology studies in nut and fruit species as well, such as walnut, chestnut, and apple, where they're finding highly correlated traits having a lot of similar QTL positions, whether they're a neighboring QTL or pleiotropic QTL, which we've seen examples of both perhaps in this data set. Pertaining to shell thickness, I mentioned we saw what would be classified as the complete range of shell thickness by the shelling industry in this population. We're seeing paper shell thin level of pecans all the way to what would be classified as a thick shell, ranging from 0.67 to 1.4, as I mentioned. Shell thickness has a direct impact on how easy it is to extract whole percent kernels or whole kernel halves, which receive the greatest price at market. And so we're looking at a very important trait here. And it's been pointed to in other studies, as Dr. Thompson and Dr. Grauke, who were two scientists out at the pecan breeding station in the past, found in a nut quality study that they did that as the shell thickness increased in pecan, the shelling efficiency and the percentage of whole kernels that were able to be extracted decreased, and that shell thickness was the single best predictor of shelling performance overall. They also found that the percentage of complete kernels extracted was also negatively affected as the width and height dimensions of these pecans increased, which kind of set me back a little bit after I saw that, because I thought width and height, increasing those would be your typical go-to thought of, oh, that's beneficial since we're increasing overall volume. But it looks like it could be a detriment if you're not increasing length at the same time. But length seems to have no real effect on this. But I think this is also tying back into the fact of, remember how I mentioned that those blocky or elliptical shaped nuts in the past have been found to be really the most common or most successful cultivars. I think that's tying back into the fact that we're seeing the adequate ratio of length, width, and height to those nuts based off of that elliptical shape, both for shelling purposes as well as filling out quality measurements as well. In terms of size, we see the complete range of what would be classified by the shelling industry in terms of size as well, ranging from oversized to small. Our largest size pecans that we saw in this population were up to 12 grams per nut, which falls into this oversized category here of 55 nuts or less needed to make a pound. If we're looking at 55 nuts to make a pound, that's roughly 8.25 grams per nut, whereas we're seeing much greater values than that. As I mentioned, 12 grams per nut, giving these a very high value to them since they're so large. And then as well as we're seeing the opposite of that all the way down to 4 grams per nut on average for the smallest pecans in this population, which falls into this small category here. But being able to parse through this and having a population that shows a complete range of size and giving some regions of the genome that have to do with this could be very beneficial and hugely important, as we could hopefully give us a starting point to eventually develop a system where we're looking at marker-assisted selection to be able to screen trees for getting a rough estimate of what size or shape nuts that they would produce before they actually get to the maturity, which can take anywhere from 10 to 15 years. And anything that saves on time in terms of producing a pecan cultivar would be extremely crucial. As you can see here, releasing a cultivar of pecan tree can take anywhere from 20 to 35 years, which is someone's entire career, basically. And so looking at where we are now and going into future collections, we only have 65 trees, as I mentioned so far, that we have data for. So really not making any concrete conclusions right now. But regardless, just with the 2 years of data that we had, this population does prove very useful for hopefully elucidating nut morphology and the genetic control of that. Of course, we'll be paying attention to the stability of these QTL, hoping to see them in the same relative regions of the genome from year to year. But I think this gives us a starting point for perhaps a genome-wide association study, or like I mentioned, giving us a starting place to hopefully develop one day a marker-assisted selection system in pecan, which has huge benefits given its long juvenility period. I think other phenotypes, like more quality aspects, like kernel color and percent fill, will come to segregate in this population as well. As I did see that there was a pretty good segregation in this population, just not enough to really pass a threshold of a 95% LOD score. But regardless, there's lots of things to find out with this population, and I think it's been a really fun project. Lots of cool stuff uncovered. And so I'd like to thank everyone that's been involved with that for my journey, my committee, Dr. Patricia Klein, Dr. Robert Klein, and Dr. Monty Nesbitt, who have been great sources of mentorship and knowledge along the way. Julie McCollum, who helps in the lab of ours, showing me machinery and all that kind of stuff. And then getting to work with the USDA ARS group. They've been crucial in getting this population started, of course, and starting this project up and hopefully keeping it going into the future. And then, of course, Texas A&M and the MEPS department for housing me. And all of this was done through the funding of the USDA SCRI grant. And so with that, I'll take any questions.
So given the long maturity period of pecans, what are some methods you can use to validate your QTLs to make sure that those aren't just false positives you're observing?
Not exactly sure, but I mean, as of right now, this is just a real starting point to get the basis of if this population will be useful to hopefully uncovering morphology of pecan. As I mentioned, it's not really been studied at all. There's lots of stuff on walnut, but not really pecan. So of course, I guess the validation is seeing the stability of those QTL as going into the future is really our main source of reliability in this population. If they're in the same spots year to year, we can hopefully assume that these are legit QTL.
You mentioned that pecans have different sizes, different blocky, more round. Do you know if there's any different applications to different types of nuts? Or do people just generally prefer blocky and that's it?
Yeah, no, there's definitely different desirability in terms of size and shape. A lot of northern material needs to mature quicker, considering they experience winter up there. And so a lot of those are typically, the early maturing varieties are typically found with smaller nut size. And so a lot of the regions up there grow smaller size nuts to help with the early maturity times. And then there is different desirability based on if you sell your pecans in the shell to the wholesale market, or if you shell them ahead of time and sell just the kernels. They kind of look for different aspects pertaining to either the size or how well the shell is actually filled.
First of all, thank you for explaining why the single pecan tree in my backyard doesn't produce any fruits. My question though is, you said it's an F1 population, right?
Mm-hmm.
The trees.
Yeah.
So does that mean that the traits that you were seeing the segregation, are they generally controlled by the seeds? So the F2, the kernels are F2, right? So
No, everything. Well, yeah, I guess they would be.
yeah,
Right.
yeah.
The seeds of that one.
Yeah, so the outside would be F1, but what's inside it would be F2. So those traits are controlled by the genotype of the F2s. Is that what you are expecting?
I'm not exactly sure.
But nothing will be segregating at F1.
Well,
It's a hybrid between the two.
Well, there's a lot of pecan's very heterozygous, and
Okay.
so
Okay.
when you're looking at one, there's still a huge amount of variability solid in just that one population. Because looking at pecan, it's almost heterozygous at every locus you look at.
Okay, so the parental lines are not.
Homozygous at all? No, yeah, it's so really you're looking at heterozygous with heterozygous, so it's all kinds of combinations.
Okay,
Yeah.
So with 5 nuts per tree, is not That's what you sampled, is that?
Well, that was in 2019 where I didn't take any of those collections. I was still rotating at the time.
And what did
So-
you do in
2020,
One or whenever.
I did 32. So
Okay.
I
So even with 32, I've bagged a lot of nuts.
Mhm.
There's a lot of variation on the same tree.
There can be, but we're typically seeing a standard size and shape. But
Did you look
32
at
would
any
be-
did you look at the variation just from a
Yeah.
single tree?
Yeah, it was relatively, of course, some trees are more variable than others, but we're seeing roughly the same amount of variation for every tree population, which it's not that much of a wide range. It's pretty stable for each tree, especially when you are getting up to 32. It might not sound like a lot, but it is definitely enough to get a relatively stable phenotype for each tree.
Want to take a question from the chat?
I have a question if nobody else does.
Okay, fire.
Well, actually, I have a couple. So the morphology of the nut is to a large degree determined by the maternal parent. Is that correct? And then the cotyledons, et cetera, inside of the nut are basically filling that internal space volume up. Is that the way it's working?
Yeah, well, regarding the first question, I don't think that shape is controlled maternally because we did take that into account. As I mentioned, there was reciprocal crosses based off of which one was the maternal parent, and we didn't see a distinct. So if VC168 was the parent, the female parent, we didn't really see a distinct shape compared that was different to say when Elliot was the maternal parent. So I'm pretty sure it's not maternally inherited, but it's based off of both parents.
Mhm.
Regarding your second question, yes, it's what they call, it's the meat of the nut, is the cotyledons, which is what is being filled in that second half of the summer. So it's
So it, I guess, is a scientific matter. Has the shape of the inheritance, the pattern of inheritance, maternal, et cetera, been described in the literature for even walnut? Is that well established at this point?
not to my knowledge, the morphology inherited in walnut is also not maternally inherited. I haven't really come across much talking about that specifically. A lot of the morphology studies are looking at just the traits themselves.
Yeah, it seems like that should be, a topic for maybe for you, but at least for somebody in the future to pursue. The other thing I was kind of curious about, so you mentioned the juvenility as going from, I think, like 7 to 15 years or something like that. I assume there must be genetic variation for that trait. Is anyone looking into that? And is that something that would be desirable to shorten, or would there be a downside to having a short juvenility period?
There's definitely differences in amount of maturity. A lot of the stuff that's been typically used in the past, since say the 1950s, when intense breeding really began at the USDA station, a lot of them used what they term precocious cultivars or seedlings, which means they mature earlier than a lot of the other species or a lot of the other specific trees in pecan. Those typically have it's been shown in literature, or at least it's been stated in the literature, that a lot of the highly precocious cultivars or trees, so the ones that mature early, typically have a lot of problems with alternate bearing or overbearing as they get older. And so I don't know, there's definitely a correlation with that. As far as the intensity of that correlation, I'm not sure, but it's definitely been pointed out in multiple different sources in the literature.
So I would kind of guess, I don't know if that would be true, but if anyone recorded that kind of information on the family materials that you've got mapping data on, that would be another trait that would be pretty useful to map out for QTL, right?
Yeah. One of the parents, Elliot, is known to be a rather high alternate bearer. So it'll produce heavy one year and not the next. But as far as VC 168, I'm not sure because it's more of a, it's an actual private cultivar. It's what the term is used. It's based out of like an orchard in California. So
But you guys can follow up on this later. I'd like to thank our speakers again.